Our next speaker this afternoon, Christie. He's a biology major from hails from Barbados. So it's been fun having Remar in the herb lab. He's helped out on a variety of projects, done salamander camp. Well, last summer was thinking, okay, he wanted a project that he could take on himself, and so I decided to look for a pathogenic fungus in Barbados to see if it was infecting populations. Unfortunately, even though I was his mentor, I did not get to join him in Barbados. Still bitter about that, but maybe for future work, I'll be down there. But the title of Remar stock, Chytrid in the Caribbean, looking for Chytrium dendrobatus and Rhinella marina populations on the island of Barbados. So, Remar.
First of all, just some acknowledgments. I'd like to thank the Animal Biology Department, Dr. Cassell, my advisor, for introducing me to Chytrid research. These guys were helping with the sample collection, and Jesse Cassell, Rebecca Cassell, and Jen Ayala for work with qPCR. Globally, amphibians as animal group are facing declines because of many threats, and 33% of amphibians are in decline currently. These are just some of the declines that they're facing, from habitat loss to fires and human disturbance. What I was focusing on is the disease portion of their declines, and as you can see, the red is a lot larger than the blue, and red just means the amount of species that are actually threatened. So threatened species are more than non-threatened species right now. What pathogen that I focused on was Chytrid fungus, which is a parasitic fungus caused by an aquatic fungal pathogen. BD is that pathogen, and it causes chytridiomycosis, which is an infectious amphibian disease, which basically infects the skins of amphibians. Chytrid is one of the few funguses that research has shown is very species specific, so it's only specific to the amphibian species, so Chytrid won't affect mammals or birds or any other species. And it's one of the few species known to cause death in vertebrates. So how does BD affect amphibians? Basically what it does is it adds keratin to the skin layer of amphibians. And if you guys know anything about amphibians, they breathe through their skin. So whether it's an amphibian that doesn't have lungs, so they breathe entirely through their skin or partially through their skin. So what happens, I don't know if you guys can see this, but this is a frog that has Chytrid, and basically it's adding on layers of skin. So slowly it's suffocating itself. It has like a drooped posture, it's lethargic, and if you put it on its back, it won't be able to right itself. These are just some symptoms of Chytrid. And research has shown that BD actually impedes the electrolyte transport system in the body. So the amphibians basically die of cardiac arrest after, I think the research says it's approximately 2 or 3 weeks that they die after infection. And it affects both adults and young. But with tadpoles, they only have keratin in their mouth parts, so that's all the BD is affecting. So it kills them slower than the adults, but if they're close to metamorphosing, and as their body transforms to get more and more keratin, the BD spreads faster and faster, and they die faster when they become adults. These are just some pictures of what happens before and after BD. Frog is all nice and moist and good looking. Not so good looking. So BD actually has 2 life cycles, and it reproduces mitotically. So its first life cycle is aquatic, where the motozoospores actually swim through the water looking for a host. When they do find a host, they embed themselves under the first layer of skin, and begin to multiply and grow. After they've done growing, they would either reproduce into more cells and reinfect that host, or go back into the water and infect another host. Modes of dispersal for chytrid is frog to frog, so infected frog touches another frog, or it leaves an infected pond, it goes to another pond, transports the chytrid with that. Some research has shown that chytrid is actually transported on the legs of birds when they land in one pond and go to another pond, or through the pet trade. So you buy something from, say, Costa Rica, had infected frogs, you buy it from Costa Rica, you bring it here, it escapes in the wild, so you basically brought chytrid to Idaho. This is just a map of like the global distribution of chytrid. So chytrid is found in every continent. Yes, it's even found in the Arctic because the wood frog actually lives up in the Arctic, so chytrid has been documented in the Arctic. And Australia, Central and North America are the areas that have shown the most dramatic declines. And whether that is because they've been studied more than other areas, or if that's just because they have more populations of frogs, we don't know. Basically, the red is areas where they have positively identified BD. Blue are areas where there's no BD, but they don't have the sample size. And white is areas where there's no BD. This is just another map showing, it's just a blown up map to show you the actual conflicts. So Barbados is a small country located in the Lesser Antilles, right over there all by itself. And it's one of the smallest islands, it's only 166 square miles. And it's relatively flat in comparison to its neighbors. It's just a map of the island and the sites that I encountered my species that I was testing, and sites that I sampled those species. So Barbados has a lack of animal diversity. That's probably because it's so flat and it's out there by itself. But we do have two species of frogs, the cane toad and the Johnston's whistling frog. And these are the only species of Panura that are present on the island. So the cane toad is a very large terrestrial toad, and this is about the size that some have been reported to getting. That's about the size of a small dog. And they're native to the Americas, so Central and South America. So as I said, they were introduced to the island to combat cane beetles, which were destroying sugarcane crops. But the people that introduced them didn't do their research, because the cane beetles live at the top of the sugarcane crops, and the frogs live on the ground. So the frogs basically overpopulated the island because they're opportunists, and they have a voracious appetite. That's just a picture of a frog eating a bat that I found with Johnny's. So the methods for the research, we did a swabbing technique introduced by Hyatt et al. In 2007. Basically what you do, catch the frog, swab its dorsal sides, its ventral sides, 5 times each, swab its hands and its feet 5 times each, for a total of 40 swabs. And you have to remember to use sterile gloves each time, because you don't want to transport BD within that species. And after the swabs were collected, I brought the samples back to Idaho to do qPCR DNA extraction on them. And basically what that does, it amplified the DNA, and it showed you if there was any BD present within that sample. And if we found BD present within the first singlet run, we would run it in triplicate, just to be doubly sure or triply sure that BD was actually in that sample specimen. So this is just a multi-component plot of the graph that we would get from the qPCR. The green is just the positive control that we put in there, so it's just a sample of BD that shows us that the reaction is working. The blue is the samples that we collected in the field. So these ones that actually rise show that there's BD present in those samples. And I think I read an article that said the closer the increase for that blue to the line, it means there's more BD present in that specimen. So there's more BD in this one than there's in that one. And this red one is just another protocol that we weren't testing. So these are my results. Yes, it was pretty pathetic, but I was in the Caribbean studying, so that was not that bad. So all the samples that I collected, none were positive for BD. So that means there was this 0% prevalence, and there was a mean infection intensity. But what does that mean? That was probably because my sample size was too small. I only caught like 12 or 15 frogs. And one of the reasons for that is because people on the island have started to use a lot of chemicals around their houses. So you're seeing less and less frogs. So I think that's one of the reasons that my sample size was so small. Also, toads have been known to have a higher resistance or tolerance for a lot of diseases. So we don't know as yet if the cane toad is totally resistant to BD, or if it just has a higher tolerance for BD than other species. So for the future, I want to investigate if BD is on the Little Johnston's whistling frog, and to see if they are less resistant to diseases. Also, I want to sample the cane toad again, but with a bigger sample size of around at least 30 individuals. If I can sample 30 individuals, I can get like a 90% sample size. So I would know that BD is either 90% on the island, or if it isn't there, it's in a small enough concentration that it's not affecting the population. And I also want to do a more widespread sampling of the island. Any questions?
Are there any other islands that have been in the Caribbean and the Antilles that BD has been found so far?
Yeah, can you go back to the map? So, of the Caribbean. So yeah, BD has been found in Dominica, Puerto Rico, Montserrat, the Dominican Republic, and Cuba so far. But those are the only islands that have been tested for it.
Oh, so they found it everywhere they've tested it?
Yeah.
Okay.
But they haven't tested the entire Caribbean. Yeah.
You might have said this earlier, but are you doing a cluster sample based on habitat in wetlands, or a random sample?
It was a random sample.
Okay. But focused on just
Focus on that species. Yeah.
any terrestrial habitat?
Mhm.
Okay.
That was because the species was known to be widespread, but when I went to sample, I couldn't find a lot of species, a lot of individuals.
Do you know why the bean fungus is able to persist in the Arctic when it's got a critical temperature of 23 degrees Celsius?
No. I haven't seen any papers on that yet.
And I would say real quick that the temperature, 23 degrees is its optimal temperature for reproduction.
Yeah.
But it can survive at cold temperatures, it just can't grow very
dormant
fast.
stage.
Maybe because the wood frog can basically die and come back to life. Maybe BD has evolved along with the wood frog. So it goes dormant with the frog, and then it comes back. But that's just speculation.
Is there a cure for BD?
Actually, there's not a cure for BD in the wild, but in captive populations, they're able to control control it and treat it.